Hey everybody, welcome to chapter 13. This is all about controlling microbial growth. I thought it was appropriate to put a little joke here about, things that look clean may not be clean. So just because it looks good does not mean that it always is good. And that's where this is coming from, a bacteria laughing at a plate. So the CDC and the NIH, so Centers for Disease Control and National Institute of Health, have established 4 different biological safety levels for laboratories, and they're determined by what type of agents the laboratories are using, how easy transmission is of the agents they're using, how dangerous the disease is, and what type of work is being done. So if you look at those, down here, you're looking at things that don't tend to cause any problems. So E. coli that is not pathogenic, Staph epidermidis, stuff like that. Micrococcus luteus in general is not really dangerous. BSL-2, these are organisms that live in and on us, and so they can cause disease, but they don't tend to if you are being safe. And so in our laboratory, for our class, we use BSL-1 and BSL-2 microbes. So Staphylococcus aureus is probably our most dangerous of all of our bacteria, but it lives on us all the time. We don't use MRSA staph, we just use staph. Does that make sense? I hope so. BSL-3s, these are where you're now starting to get into microbes that tend to cause pretty dangerous diseases. And then BSL-4, those are a lot of the viruses that are fatal and microbes that are fatal, that cause very dangerous. Not that Mycobacterium tuberculosis, which causes tuberculosis, isn't fatal. It is, but it's not as deadly as Ebola. So before we get into how we control microbial growth, there are a few terms I always like to go over. The term fomite is an inanimate item, so a doorknob, a handkerchief, a piece of clothing, that can harbor microbes, and then if you touch them, you can get the microbes. So think about if you're cleaning up your house and say your significant other or your sibling or parent, depending on who you are and where you live, has a cold and they're blowing their nose, and then they leave their balled up Kleenexes sitting on, say, the coffee table. Now you're picking those up. Those Kleenexes are called fomites because they actually contain the pathogen that caused the common cold, and they can transmit it to you, which is why you should always tell them they need to clean up after themselves. But a glass, if you drink out of a glass, that can also be a fomite. If you touch a door handle, if you use someone else's phone, all of those are fomites. The term disinfection then is the removal of, in general, the potential pathogens. But when you disinfect, you're not getting rid of all microbes. It's not a sterilization process. Disinfectants in general are used on non-living surfaces. So we often disinfect like silverware through a process known as sanitation. If you worked at McDonald's, so I use McDonald's because that's where I worked. That was the one restaurant I worked in. We would have to clean our dishes. We would wash dishes in hot soapy water, rinse them, and then we would stick them in a sanitizer solution, which was a warm solution that contained a special set of chemicals with chlorine. They'd sit in there for like 5 minutes, and then we'd take them out. And that was to make sure that they were deemed acceptable for safety standards. An antiseptic then is a type of disinfectant used on living tissue. So things like betadine is considered an antiseptic. The term sterilization then is the removal of all life. So something that is sterile has no microbes at all, no endospores, no viruses, nothing that, so not just life, the removal of all microbes. It's clean of everything. So how do we determine what type of method we're going to use to control microbial growth? There's going to be a lot of different factors that play into this. What type of organism that we're dealing with, how many of those organisms we have to deal with, the environment that we're trying to control the microbes, the risk of infection, and the composition of what we're trying to clean. So when we talk about type of organisms, some organisms are more resistant than others. So here we have endospores, and here we have protozoan cysts or oocysts. Both cysts and oocysts, as well as endospores, can persist in environments that are not beneficial to life. And because of that, if we put chemicals onto endospores, they're not going to likely some chemicals, I'll say it that way, aren't going to destroy the microbes. And so you have to use something else if you're dealing with endospores or protozoan cysts. Another example of an organism that is resistant is Mycobacterium. Because of the mycolic acid in the cell wall, it's going to be very resistant to certain chemicals. And then there are some organisms that actually grow in disinfectants. Example is Pseudomonas oftentimes will be growing in a betadine solution. The number of microbes. So how many microbes are in the environment that you're trying to kill off? If there's a lot of microbes, it's going to take longer time than if it's a small population. When I'm using my autoclave, if I'm trying to sterilize media that has not very much microbes likely in it, because I haven't inoculated anything, I use a 15-minute time period. But if I'm trying to get rid of biohazard, so you guys have run a lot of tests, and I have, a couple biohazard bags full of media. If I'm running those, it's going to take longer to make sure that everything gets killed. So how do microbes die? The process of killing microbes, they actually die in a process known as decimal reduction. So they are dying in a negative logarithmic pattern. So they're dying exponentially, but they die slower than they replicate. That is a truth. And we're going to talk about that in a little bit, what decimal reduction time means, okay? So then we're looking at the conditions. So what type of environment are we using these chemicals, or are we going to use whatever treatment we're using? If the conditions contain a lot of organic matter, then the material may not be as effective. So it's always important to clean the environment before you actually try to disinfect or sterilize the environment. Think about if you spray Clorox cleaner on your kitchen counter that has a bunch of blood and fluid from the chicken, the raw chicken that you opened up. If you didn't wipe that up first, then the likelihood of Clorox actually killing all the salmonella would be very slim. But if you clean up the mess first, make the counter clean, then you spray the bleach on there, the Clorox cleanup bleach, let it sit for the 5 minutes it needs to sit, and then wipe it off, then you're going to kill those cells. And then the risk of infection. So what this is looking at is if it's something that is going to be a critical risk for infection, you need to have it sterilized. If it's semi-critical, then you still want to make sure that it is sterile. It's not as at risk, but you definitely want to, you're not going to use the same endoscope on 2 people. If it's non-critical, then you might be sanitizing it. So think of like stethoscopes or blood pressure cuffs. You're going to reuse those. Whereas an endotracheal tube, you're not going to reuse. And needles, you're not going to even sterilize. You're just going to get rid of them. The composition, so different media need different treatments. You can't heat plastic because plastic or other heat labile materials are going to break down in heat. Irradiation can be used, but it can also damage some things. And moist heat can be effective at cleaning certain things, but there are certain materials like steel that is moisture sensitive. So you're going to have to choose the treatment based on what type of item you're trying to clean. All right, so I told you that we were going to talk about decimal reduction time, or how we kill microbes. So microbes are not killed instantly. It's not like you spray bleach on them and they all die. Boom, we're dead. They die the same way that they live, or the same way that they grow. And they grow exponentially. They die exponentially. But the death rate is slower in time than their growth rate in general. So you don't see this really fast growth rate where it goes straight up. You don't see a fast death rate going straight down. You see more of a smooth death rate going at an angle. But decimal reduction time is the time it takes to kill microbes, 90% of the microbes within a given population at some given temperature. And so it's usually written as the D with a temperature in Celsius next to it, okay? So if you start out with 10 billion microbes, okay, so that's right here, 10 to the 10th. And if we're using something that kills microbes, so that has a D time of 5 minutes at some temperature, then every 5 minutes we're killing 90% of that population. And so the way to figure out the D time, you can do it this way, where you have 10 to the 10th. And so you can count these. There's 3, 6, 9, 10. So 10 to the 10th number of cells is 10 billion. And then we reduce the decimal each time. So then it'd go 10 to the 9th, 10 to the 8th, 10 to the 7th. I always just say move the decimal over each time. And after a certain number of D times, you're going to have less than 1 full cell. So here at 5 minutes you have 1 billion cells. At 10 minutes you have 100 million cells. At 15 minutes you have 10 million cells. At 20 minutes you have 1 million cells, then 100,000 cells, then 10,000, 1,000, 100, 10, and at 50 minutes you have 1 cell left. If you stop the treatment at that point, that single cell can replicate and produce all kinds of new cells. So you had to go for 5 more minutes to get less than 1 full cell, and then you kill off all the bacteria. So I'm going to stop this video here, and I want you guys to try and figure this out. And then in our next video, I'll go over it, okay? All right, bye.